Hello, my name is Joshua Mugabe. I welcome you to Osmoregulation in Paramecium. Paramecium and amoeba live in fresh water, and their cytoplasm contains a greater concentration of solutes than their surroundings. So, they absorb water by osmosis. The paramecium and the amoeba, they live in fresh water, but they have a high concentration of solutes inside their cytoplasm. There is a high concentration of salts inside their cytoplasm compared to the solutes which are in the water or in their surrounding. There is less concentration of solutes in their surroundings. So what is going to happen is that now water is going to move from a region of low solute concentration to a region of high solute concentration. So water will move from the surrounding and it will be entering into the cell of the paramecium and the cell of the amoeba by a process of osmosis. So water will be moving from a region of high concentration of water to a region of low concentration of water, or a region of low concentration of solutes and a region of high concentration of solutes. That's how water is going to move, by the process of osmosis. The excess water is collected into a contractile vacuole, which swells and finally expels water through an opening in the cell membrane. So what is happening is that this water is going to enter through, or will enter into, the contractile vacuole through the radiating channels. And here we have one of the vacuole, as you are seeing here. It is filling up. It is small. And this vacuole that we are seeing here is already full with water. It's ready to be emptied. So the water is entering into the contractile vacuole, and when it enters into the contractile vacuole will start to swell. And later on, this swollen contractile vacuole will open at the cell membrane and release this excess water outside of the cell. In amoeba, the vacuole forms near the posterior end, but is not associated with a particular structure. When you look at the amoeba, you will see that it has a contractile vacuole here. And when you look at that contractile vacuole, it is near the posterior end, and it does not have any structure associated with it. And although it empties its content through an opening in the cell membrane, this also does not appear to be a permanent pore. So when this contractile vacuole is filled with water, it is going to empty its content through a pore, and it releases the content outside the cell membrane. But this pore is not permanent at that particular point. Another time it may release it from here, another time it may release it from this side. So meaning that the pore where it releases the content from the vacuole to the outside, that pore is not permanent. In the paramecium, for it has two contractile vacuoles, one at each end, and they fill and contract alternately. So when you look at the paramecium here, you are going to see that the paramecium has got two contractile vacuoles. One is here and another one is here. So there are two contractile vacuoles at both ends, and they are filling. This one you can see it is just filling. This one here, the contractile vacuole is small because it is just starting to fill. Whereas the contractile vacuole this side is already filled up. You can see it is swollen and it is ready to expel the water outside. So they are going to contract and fill alternately. When this one is filling, this one is contracting. And when this one now has released its content out, now this one will be swollen. And then it will also release the contents later on. So that's what is going to happen there. They are permanent structures, and supply water is conducted to the vacuoles by a series of radiating channels. Now this one is with the paramecium. When you look at the paramecium, it has a contractile vacuole on both ends of the cell. And you'll see that for it has other organelles attached to the contractile vacuole. The contractile vacuole here you are seeing inside, it is having other organelles which we are calling the radiating channels attached to it. So we have the radiating channels attached to the contractile vacuole, and it is going to supply water to the vacuole, and this water is going to pass through the radiating channels. It passes through the radiating channels to enter into the contractile vacuole. And we have seen that these structures are permanent. One structure is at the posterior and another structure is at the anterior. So that's how they are going to be moving, because for them they are just permanently situated or placed in particular positions. Hello, my friends. Thank you very much for participating in osmoregulation in paramecium. Please do like, share, and subscribe.